Today's lecture looks at the properties and functions of muscles. The content of today's lecture is covered in Chapter 3 of the Newman textbook. Muscles have multiple functions. Typically, we only tend to think about one function, that is, the ability of a muscle to produce a movement. In this sense, the function of a muscle is to act like a motor and to power the movement. Muscles can also perform the opposite function. They can act like brakes to suck energy out of a movement. When you're walking down some stairs next time, pay attention to how your muscles are acting to brake your fall as you step down each step. Muscles can also act to stiffen a joint and turn two body segments into one rigid segment. Muscles can act to maintain body posture. They can act as structural supports and to protect bones and internal organs. They can control pressure, such as in the case of breathing and speech. They can support the regulation of body temperature, and they are needed for the intake and excrement of solids and liquids. Muscle has 4 key properties that allow us to contrast it with other types of tissues in the body. These properties are irritability, contractility, extensibility, and elasticity. Irritability refers to the ability of a muscle to respond to a stimulus. Contractility refers to the muscle's ability to shorten in length. Extensibility refers to a muscle's ability to extend in length. And elasticity refers to the muscle's ability to stretch and return to normal length. We're going to briefly consider 3 factors that affect the amount of joint excursion that is possible when a muscle is crossing a joint. The first factor is muscle length. Muscles with longer fibers produce greater excursion than muscles with shorter muscle fibers. Greater length is related to having more sacromeres. The second factor that affects the amount of joint excursion that is possible when a muscle is crossing a joint is muscle morphology. For example, fusiform or strap muscles permit more joint excursion than pennate muscles. Here we see examples of muscles with various different morphologies. The third factor that affects the amount of joint excursion that is possible when a muscle is crossing a joint is moment arm. Muscles with shorter moment arms will produce more angular excursion than those with larger moment arms that have similar length. Here we see the biceps muscle illustrated with its normal insertion point on the radial tuberosity, as well as illustrated with a hypothetical insertion point that is further away from the axis of rotation. Given a fixed shortening ability of the biceps, if we move the insertion point further away from the axis of rotation, the amount of possible joint excursion will decrease dramatically. So what is the clinical application of knowledge about the factors that affect joint excursion? One application is in determining donor muscles for tendon transfers. For example, in radial nerve palsy, finger extension can be restored by weaving the flexor carpi radialis into the extensor digitorum. The flexor carpi radialis is chosen because it has similar shortening ability. Here we see an illustrated example of a tendon transfer procedure used for restoring wrist extension. In this operation, the pronator teres is woven into the extensor carpi radialis longus. So what are the factors that affect the strength of a muscle? The force that can be exerted by a muscle depends upon the size of the muscle, pennation angle, by that I mean the directions in which muscle fibers are pulling, muscle moment arm, muscle stretch, contraction velocity, neuromuscular mechanisms, and fiber types. Let's consider the factors that affect muscle strength one by one. Speaking loosely, more muscle bulk is associated with greater muscle force production. More specifically, muscle force is proportional to a greater number of cross bridges, it's proportional to a greater number of actin-myosin filaments, and it's proportional to a greater number of muscle fibers. Muscle size is often evaluated in terms of muscle cross-sectional area. Two measures of muscle size are physiological cross-sectional area and anatomical cross-sectional area. Physiological cross-sectional area and anatomical cross-sectional area are not always the same thing. Here we see a depiction of anatomical cross-sectional area with the blue line and physiological cross-sectional area with the green line. Anatomical cross-sectional area in blue is the area of the cross-section of a muscle perpendicular to the longitudinal axis of the muscle. In contrast, physiological cross-sectional area is the area of the cross-section of a muscle that is perpendicular to its fibers, generally at its largest point. The meaningful difference between anatomical and physiological cross-sectional area is revealed when we look at these measures across different muscle types. The physiological cross-sectional area is much greater than the anatomical cross-sectional area in pennate muscles compared to fusiform or strap muscles. The strength of muscle depends upon pennation angle. Pennation angle refers to the angle between the orientation of the muscle fibers and the orientation of the tendon. We can see the orientation of the tendon with the black line and the orientation of the muscle fibers with the red line. A typical pennation angle is 30 degrees. So, what do pennation angles mean for the forces experienced by tendons? The tensile force exerted by a muscle on a tendon is equal to the cosine of the pennation angle multiplied by the force exerted in that direction. In our picture here, if the pennation angle that we're seeing is 30 degrees, this means that only 86% of the force produced by the muscle will be acting to pull the tendon. Muscle strength also depends upon the amount the muscle is stretched. The forces that are transferred from one bone to another during a muscle contraction depends upon the elastic properties of multiple tissues. The tension that is generated depends upon the passive elastic contractile properties of tendons, the passive elastic properties of connective tissues within the muscle, and the active contractile properties of elements within the muscle. As we just mentioned, the forces generated by a muscle depend upon the passive elastic contractile properties of muscle tissues. The passive forces that are generated when a muscle is stretched are revealed with in vitro testing of muscle lengthening. In vitro testing is when muscles are removed from the body and tested outside the body. Studies of muscle lengthening in vitro reveal a passive length tension curve that shows how elongating the limb to stretch a muscle will exert a passive tension in the muscle. So here we can see results where a muscle is removed from the body and is slowly stretched. We can see that the fibers in the muscle are initially slack, and as the muscle is stretched until that slack is taken up, it's not going to generate any force. Then, once the fibers become a little bit taut, we start to develop passive tension. So, the further we increase the muscle length, the greater tension we see building up. These results show that simply elongating the limb will generate a passive tension in the muscle. We just looked at the passive length tension curve, the graph that shows the build-up of tension as a function of passively stretching the tissues. Let's now think about the curve that shows how force is built up when we combine both active and passive elements. So, here we have the active-passive length tension curve. We can see the active forces shown in the grey line here, and we can see the passive forces with the red line here. and we can see the total force generated being realized as the combination of these two forces adding together. The results that we get from in vitro testing of muscle fibers reveal that when we stretch muscle tissues far beyond its resting length, we get a large amount of forces. So we can see the black line coming up here, and as we get to stretch the tissues really far, we get some really high forces created. Something to bear in mind when you're looking at this data is that in vivo, when the muscle is actually acting in the body, these lengths are rarely experienced. So these tensions are very rarely created. When we consider a muscle in vivo, that is, embedded within the complex tissue network of the body, we see multiple force transmission routes. The major force transmission routes are myotendinous and myofascial. Myotendinous force transmission involves forces transmitted from tendon to muscle to tendon. Myofascial force transmission involves forces transmitted from tendon to muscle to adjacent muscle through connective tissue to adjacent tendon. In some cases, it has been shown that as much as 50% of the force transmitted through the body goes through myofascial routes. Here we see a diagram of the elements of muscle stretch. The basic functional unit of the active contraction of muscle is the muscle fiber. To understand the mechanism of muscle contraction, we need to look at what is going on within muscle fibers at the level of the interactions between myofilaments. The sacromere provides the most basic unit of active force generation. At the top, in this picture, we see an electron micrograph of two full sacromeres. In this micrograph, the three vertical black lines are the Z-discs. Z-discs are the anchor points for actin filaments that are shown in red in the diagram of the sacromere structure at the bottom of the picture. The actin filaments overlap with myosin filaments. As we will discuss next, muscle shortening occurs through a molecular ratcheting mechanism that pulls the myosin filaments over the actin filaments. Shortening of many sacromeres in unison is what creates muscle movement. Our understanding of the ratcheting process that pulls actin filaments over myosin filaments is called the sliding filament theory. Here we see a diagram representing the action that occurs at a single point of contact between an actin and myosin filament. The action happening here is called cross-bridge cycling. In cross-bridge cycling, a myosin filament head molecularly attaches to the actin filament. The myosin filament head then changes shape, which acts to pull on the actin filament, and then the myosin head detaches and returns to its original shape. We can understand this process in even greater detail by considering what is known about the mechanochemistry of the myosin filament head. When an adenosine triphosphate or ATP molecule binds to a myosin filament head, it acts to cock the head in an extended position. When the myosin head comes in contact with the actin filament, a power stroke occurs, in which the myosin filament head pulls itself back into its original shape. With a more detailed understanding of the mechanism of active muscle contraction in hand, we can start to add some interpretation to the active length tension curve we were looking at a little bit earlier. Here we see a graph of the tension that's developed as a function of the length of a single sacromere. Remember, the sacromere is the basic unit of muscle contraction, consisting of an actin filament and a myosin filament. Here we see 4 different sacromere lengths. In the arrangement labeled D, the sacromere is so stretched out that there is no overlap between the actin and myosin filaments, and no cross bridges can be formed. In the arrangement labelled B, the maximum number of cross bridges can be formed. In this arrangement, we can see that the maximum amount of active force can be generated. In the configuration labelled A, we see that the sacromere has contracted so much that the actin myofilaments are starting to overlap. This overlap leads to interference and a reduction of the number of effective cross bridges that can be formed. To sum up, what we see here is that changes in the proximity of actin and myosin filaments influences the potential force that can be generated in sacromeres. Maximal stretch or maximal contraction both are not optimal states for generating muscle tension. Maximal force occurs when the maximum number of possible cross bridges are possible, and this occurs at the resting length of the muscle. Let's go through this one more time, this time with a graph of the length of a muscle on the x-axis and the isometric tension generated by that muscle on the y-axis. Changes in the proximity of actin and myosin filaments influences the potential force of the muscle contraction. When a muscle is at its resting length, a maximum number of cross bridges can be formed. This corresponds to a sacromere length of about 2.5 micrometers. In lengthened muscle, the length of the sacromere is about 4 micrometers. In this configuration, the number of cross bridges that can possibly be formed is considerably reduced. This means that less force can be generated. Lastly, in a shortened muscle, the sacromere length can reduce to about 1.5 micrometers, and overlapping cross bridges can lead to interference and a reduction in the number of effective cross bridges. Let's now think about the case of muscles that span more than one joint. Multi-joint muscles tend to stretch more than single joint muscles and have higher tension at longer lengths. Thinking about muscles that span multiple joints has a number of implications for how we understand the function of muscles. Multi-joint muscles create the possibility of multi-joint force generation strategies. Take the hamstrings that we see illustrated. If you bring the hip into flexion by sitting in a chair, the hamstring that spans the hip and the knee will be tightened. It will be passively tensioned. If you then perform a knee flexion, the force you will be able to produce will be increased. Similarly, if you bring the hip into flexion by raising your knee up in front of you when you are laying down, the hamstring that spans the hip and knee will be tightened. It will be passively tensioned. If you then extend the knee, you will create significantly more stretch. Let's think for a moment about some of the clinical implications of multi-joint muscles. One simple example is hamstring injuries. You can expect to see a person walking around with a hamstring injury to be walking with a flexed knee in order to avoid tensioning the injured tissues. Another important clinical example relates to strength and range of motion testing. If you want to evaluate joint strength, there are multiple options for how you could choose to do it. For example, on the left we see knee flexion strength being evaluated in a laboratory using isokinetic dynamometry. On the right we see knee flexion strength being measured with a clinician holding a handheld dynamometer. There are many reasons that we might expect these two different measures to yield different results. But for now, I just want you to think about the effect of multi-joint muscle action. I will leave that question with you. The relationship between the angle of a particular joint and the torque that can be produced at that joint varies across muscle groups and is related to the functional demands on that particular muscle group. For example, if we look at the torques that can be produced by the action of the elbow flexors in different elbow postures, we see that the torque potential is greatest in the anti-gravity posture. In other words, in the posture where the external force on the limb is likely to be at its greatest, we see the involved muscles having their greatest potential to create internal forces to balance the external forces imposed by gravitational forces. If we alternatively look at the hip torques that are produced as a function of hip angle, we see a very different relationship. Here, the greatest potential for torque generation occurs around a neutral hip posture. This posture is adopted in the stance phase of gait, when large abduction torques are required for pelvic stabilization. As we will discuss shortly, it's important to realize that we're talking about joint torques on this slide, and not muscle forces. This means that the values that we're seeing in these graphs are not only due to the active forces generated by the elbow flexors and hip abductors, respectively, they're also non-trivially determined by the effects of muscle moment arms. The torque that can be created at a joint depends both upon the length of the muscle and the moment arm. To understand this, let's look at the torque produced by the biceps brachii. Here we see the data for the torque produced by the biceps as a function of elbow angle. If you compare the pattern of this data to the graph for elbow torque on the last slide, you will notice that they are similar. This is data from subjects performing a maximum voluntary contraction. We see that the torque produced by the biceps is greatest in the anti-gravity posture, just as we saw on the last slide. The torque that we're seeing here depends upon the interaction of muscle length dependent changes in the forces produced by the biceps and joint angle dependent changes in the moment arm of the biceps muscle force vector. When we look at the graph of muscle force versus muscle length, we see that the greatest force is generated when the biceps is near its maximum length. When we look at the moment arm of the biceps muscle force vector, we see that the moment arm is largest when the arm is at 90 degrees of flexion. These factors combine to produce the biceps torques that we observe. This is just the case for the biceps. The relative contributions of muscle moment arm and muscle length to the torque that is produced at a joint varies with each muscle and joint. There are some basic clinical implications of the interactions between muscle length and muscle moment arm in joint torque generation. During evaluations, it's important to keep joint positions the same when testing muscle strength pre- and post-treatment. Having constant joint positions for both pre- and post-treatment evaluations will control for posture-specific changes in the muscle length and muscle moment arm. For example, if you measure hip flexion strength while a patient is sat in their wheelchair pre-treatment, and then measure hip flexor strength when the patient is laying down after the treatment, how will this affect your conclusions about the effectiveness of the treatment? Again, as I did earlier, I will leave this question with you to think about. The next factor to consider when attempting to understand the force and torque generating capacities of muscle is velocity. Here we see the forces generated during a maximum effort muscle activation as a function of contraction velocity. The zero value on the x-axis represents zero velocity. Thus we are looking at an isometric or constant muscle length contraction. In an isometric contraction, the maximum number of cross bridges are given time to form. The values on the right side of the graph are for when the muscle is shortening while it's being innervated. Activation of a muscle during muscle shortening is referred to as a concentric contraction. What we see here is that since the muscle and its constituent sacromeres are shortening, the time allowed for actin-myosin cross-bridges to be formed is reduced. As the velocity of the concentric contraction is increased, fewer and fewer cross-bridges can be formed, and the force that results gets lower and lower. The values on the left side of the graph are for when the muscle is lengthening while it's being innervated. Activation of a muscle during muscle lengthening is referred to as an eccentric contraction. Here we see that the muscle force increases with the rate of muscle lengthening. Referring to actin-myosin cross-bridges again, we can interpret this as greater muscle force results from added force from cross bridges being pulled apart and detached, more rapid reattachment phases of cross bridge formations, and added tension from passively stretched viscoelastic muscle components. You will notice that a plateau is observed on this side of the graph. This is potentially due to limits on the ability to form and reform cross bridges at higher muscle lengthening velocities. There are some basic implications to be drawn from the effects of contraction velocity that we looked at on the last slide. Maximum eccentric contractions are 1.5 times stronger than maximum concentric contractions. Eccentric contractions are thought to contribute to delayed onset muscle soreness. This soreness is believed to be associated with small-scale microtraumas to the muscle fibers. The last factor that we're going to consider that contributes to understanding the strength of a muscle is neural activation. The basic functional unit of neural activation is the motor unit. The motor unit consists of a single motor neuron and the muscle fibers it innervates. The nervous system recruits a motor unit by altering the voltage potential across the membrane of the cell of the motor neuron. At a critical voltage, an electrical signal is created. This is referred to as an action potential. This action potential is propagated down the axon to the motor end plate at the neuromuscular junction. The individual muscle fibers that are activated by the action potential contract. A contraction of a single muscle fiber is referred to as a twitch. Muscle force production results from the activation of a large number of motor neurons and the contraction of the many muscle fibers that they innervate. Motor units differ in size. All have just one motor neuron, but in large motor units, the motor neuron connects to many muscle fibers, while in small motor units, the motor neuron connects to only a few. The size of motor units is important for understanding how force builds up in a muscle. Small motor units tend to be recruited before large motor units. This is referred to as the size principle. A consequence of the size principle is that it allows force build-up to happen smoothly, without large jumps in force. Another relevant characteristic of motor units is that each action potential results in a contraction that only lasts a fraction of a second, specifically somewhere between 100 and 300 milliseconds. This means that maintaining a muscle force requires repeated excitations of each motor unit. The second principle that's important for understanding how force builds up in muscle is rate coding. The force produced by a muscle can be controlled by varying how frequently muscle fibers are contracted. Here we see a graph that illustrates the buildup of muscle force over time as a function of the contributions of 4 different motor units. Here we see the firing patterns associated with the 4 motor units. Each vertical line represents a single action potential occurring in one of the motor units. You can see that the height of the line increases as we go from motor unit 1 through to motor unit 4. This is illustrating that the size of the motor unit is getting larger as we go from motor unit 1 through to motor unit 4. Remember that the size of a motor unit relates to the number of muscle fibers that are innervated in that unit. Motor units have been found to innervate as few as 5 muscle fibers and as many as 2,000. We see then that motor unit 1, the smallest motor unit, is recruited first. Motor unit 4, the largest motor unit, is recruited last. Let's focus on the recruitment of motor unit 1. The first time it fires, we see a small bump of force created. This then fades away before the motor unit is activated again. As the rate of firing of the motor unit 1 increases, the contraction forces created start to overlap and force starts to build up. In general then, what we see is that any newly recruited motor unit will fire at a base frequency, and then the firing rate will increase to a maximum. Before the maximum possible firing rate for Motor Unit 1 is reached, we see Motor Unit 2 being recruited. Now the effects of Motor Units 1 and 2 are being combined to produce the observed muscle force. Muscle fatigue concerns the decrease in muscle force over time. A decrease in the ability to produce muscle force over time can result from either central or peripheral sources. Peripheral fatigue is produced by changes at or distal to the neuromuscular junction. Peripheral fatigue depends upon the proportions of different motor unit types. If a muscle has a high proportion of fast fatigable motor units, it will fatigue more quickly. Central fatigue concerns processes within the central nervous system that reduce neural drive to the muscle. Evidence of central sources of fatigue come from studies that show that faster muscle fatigue occurs in different tasks, even though similar tasks are required for those tasks. Other evidence of central influences on muscle force production come from brain imaging studies, and from studies that show that mental rehearsal can actually increase the force that can be produced by a muscle. Potential sources of central fatigue include changes in motor cortex excitability and the influence of afferent inputs at the spinal level. An important clinical implication of the mechanisms of muscle fatigue is that rest periods should be varied depending upon the particular muscles exercised. The important characteristic here is the specific composition of the different motor unit types that exist within a specific muscle. A muscle that is rapidly fatigued by high intensity and short duration exercise can recover after a rest period of only several minutes. In contrast, a muscle that is fatigued by low intensity, long duration exercise usually requires a much longer time to recover its force generating capacity. Here we see some data from an experiment in which participants repeatedly isometrically contracted their elbow flexors. Each contraction lasted for 6 seconds, followed by 4 seconds of rest. In this task, participants produced 5 contractions with 50% of their max force. Then they produced 1 contraction with 100% of their max force. This pattern was then repeated. In the case of the 50% max contractions, only a subset of the motor unit pool is required for each contraction. This means that as some motor units get fatigued, there are still unfatigued reserves to draw upon. Thus, as time goes by, and some of the pool of motor units gets fatigued, you see a greater and greater number of motor units being recruited to produce the required force. This is evident in the increasing magnitudes of the EMG signal, which is shown in red. The EMG signal is showing the increasing level of recruitment that is including more and more motor units as time goes by. In the case of the 100% max contractions, the participant draws upon the whole pool of available motor units from the outset. So, as some motor units get fatigued, there is a drop in force. So if we look at the force graph here, over time we see that the amount of force decreases over time. What we're seeing in this data is redundancy in the motor unit recruitment for submaximal actions. As soon as some motor units get fatigued and are unable to produce a force in response to an activation potential, the participant must call on other motor units to get the job done. Here we see that more and more motor units are being recruited in order to achieve the same output. What are the muscle strength related adaptations we observe following exercise? The effect of exercise that we are all familiar with is muscle hypertrophy. Hypertrophy results from increased protein synthesis within muscle fibers and therefore an increase in the physiologic cross-sectional area of the whole muscle. Protein synthesis results in sacromeres being added in parallel within the muscle fibre. Hypertrophy is greatest in type 2, or fast-twitch fibres. Other exercise-related adaptations that impact muscle strength include increased discharge frequency of motor units, increased motor neuron excitability, increased motor cortex activity, and decreased inhibition at spinal and supraspinal levels. So what do the effects of exercise on muscle look like? In one study, the effect of high-resistance training was investigated. This study involved concentric and eccentric contractions performed 3 times a week for a 12-week period. The study revealed a 30 to 40% increase in 1 rep max. In weight training, 1 rep max is the maximum amount of weight that a person can possibly lift for 1 repetition. One of the most dramatic responses to strength training is muscle hypertrophy. In another study, 20 weeks of high-resistance strength training was shown to result in a 30% increase in muscle cross-sectional area. Since eccentric activations produce greater force per unit of muscle, eccentric training can be more effective in promoting muscle hypertrophy. Reduced muscle use, or muscle disuse, can occur following a trauma that requires a person's limb to be immobilized, or when a person is confined to bed due to illness or disease. Loss of strength can occur early, up to 3 to 6% per day in the first week alone. After only 10 days of immobilization, healthy individuals can experience up to a 40% decrease in 1 rep max strength, and up to a 20% reduction in fiber cross-sectional area. Protein synthesis is reduced in all muscle fiber types when you have a chronically immobilized limb. Most notably, this occurs in slow-twitch or type 1 fibers. This is related to the fact that slow-twitch muscle fibers are used to being recruited frequently throughout daily activities. Relatedly, postural or anti-gravity muscles that have predominantly slow-twitch type 1 fibers show more rapid atrophy. Let's talk about adaptations in muscle in response to aging. After the age of 60, we begin to see a reliable loss of strength as a function of increasing age. Strength is lost at a rate of approximately 10% per decade. The loss of muscle tissue, or sarcopenia, is connected to the progressive loss of alpha motor neurons and the loss of fast-twitch type 2 fibers. The rate of muscle loss is muscle-specific. For example, the loss of strength is generally more pronounced in the muscles of the lower limb, such as the quadriceps, as compared to muscles in the upper limbs. There is a reliable relationship between muscle strength and the ability to perform functional activities. Strength definitely doesn't determine the functionality, but it is a very important contributing factor. Repeatedly in the literature, it has been shown that interventions that target improvements in strength can have a positive impact on functional abilities. A basic take-home message of such findings is that strength is important to measure in geriatric populations. For the final part of this lecture, I would like to talk about electromyography. Electromyography, or EMG, is the science of recording and interpreting the electrical activity that emanates from activated skeletal muscle. In activated skeletal muscle, electrical impulses are carried down the alpha motor neurons of a motor unit to the motor end plates. Activity then propagates in both directions along the length of muscle fibers. The sum of all of this electrical activity is evaluated via EMG. Electromyography is performed using electrodes to measure the electrical activity in activated muscle. Electromyography typically employs either indwelling electrodes or surface electrodes. Indwelling electrodes record electrical activity by inserting fine wires into the body of a muscle. In contrast, surface electrodes are placed on the surface of the skin and are positioned over an underlying muscle of interest. There are 3 common types of EMG sensors that use surface electrodes. The most basic type of surface EMG simply involves metal discs stuck to the skin, and wires that connect those discs to external electronics. The external electronics measure and interpret the electrical activity induced in the discs. Using this method of surface EMG requires that 2 electrodes are replaced over the muscle belly of interest, and 1 reference electrode is attached to a part of the body where minimal electrical activity is occurring. This means trying to position this reference electrode over bones rather than muscles. Other EMG sensors have the electronics that measure and condition the electrical signals from the electrode built into them. The most advanced forms of EMG sensors are wireless electrodes that also have the integrated electronics that condition the signal built into them. The basic theory behind electromyography is as follows. We have 2 surface electrodes placed on the skin over a muscle belly of interest. We also have a reference electrode placed over a bony region on the body. These 3 electrodes are connected to electronics that are designed to measure the electrical activity. The first part of the electronics is referred to as monopolar EMG. Each of the red triangles here represents electronics that are connected to a surface electrode and to the reference electrode. These electronics measure the difference in electrical activity between the reference electrode, where we expect no activity, and the surface electrode, where we expect to see the electrical activity of activated muscle beneath the electrode. Given how tiny these voltages are, the other thing that's done here is to amplify the signal. The 2 amplified monopolar EMG signals are then fed into the blue triangle. The blue triangle represents electronics that take the 2 amplified monopolar EMG signals, measures the voltage difference between them, and then amplifies the result. The function of these electronics is to remove unwanted external electrical interference from the measurement. Lastly, the bipolar EMG signal is sent to more electronics that convert the analog voltages that are being measured into digital signals that can be interpreted by a computer. Whenever you are using any kind of test or measure, the first question that you should always ask is, what am I actually measuring? What is this test or measure actually telling me? With EMG, given that you are measuring the electrical signals that trigger active skeletal muscle, it's quite tempting to assume that EMG is a measure of how much force is being generated in the muscle. Unfortunately, this is not exactly the case. EMG is never a perfect measurement of exactly what the muscle is doing. No matter what technique or what technology you're using, there is not a way to measure all of the activity in a muscle. This means it's not possible to know how many muscle fibers are currently being contracted. So let's consider this issue. So here we see 2 electrodes placed on the surface of the skin above a group of muscle fibers. We also see 2 alpha motor neurons that are innervating these fibers. Notice that we have numbered the muscle fibers based upon how close they are to the electrodes, with the muscle fiber labelled 1 being the closest to the electrodes. Here we see a representation of the electrical signals from each of these muscle fibers that would be measured by the EMG sensor, if each of them were to fire individually. Even though the electrical activity propagating down each of these muscle fibers should be the same, the EMG will measure the electrical signal that is generated by the muscle fibers that are closest to the electrodes as being much stronger than the signals from the muscle fibers that are further away from the electrodes. When electrical activity is present in all of these muscle fibers at the same time, the EMG signal only shows the combined activity. This means that if one muscle contraction recruits muscle fibers that are close to the electrodes, and another muscle contraction recruits the same number of muscle fibers, but those muscle fibers are further away from the electrode, it will seem like there is more muscle activity in the first recording. So, knowing these limitations, what does an EMG signal tell you? The signal is going to be related to active contractile force generation. But the EMG sensor is only measuring part of the activity in the muscle. This means that while the measure is correlated with muscle force, it's often not a reliable measure of muscle force. In contrast, EMG does seem to be a good way of measuring the onset of muscle activity. As long as the sensor can measure some of the motor units that are being recruited for a given action, it will be possible to know when an active muscle contraction is initiated. This type of method is important for assessing the functionality of muscle recruitment patterns. Making an EMG recording is relatively straightforward. Let's take a look at the simple task of recording the activity of the biceps. To make sure that measurements of the muscle can be compared across sessions, care should be taken to position the electrode over the same part of the muscle for each test. This will ensure that the same grouping of muscle fibers and motor neurons in the muscle are being measured to a reasonable degree. Ideally, electrodes should be placed over the muscle belly to get the clearest signal, and the two electrodes should be lined up along the direction of the muscle fibers of the muscle that you're looking at. For the biceps, the electrodes should be placed close to the bulk of the muscle in mid-arm. The recommended test position for this muscle is to have the patient laying supine with their arm extended out to the side. The recommended test maneuver for making sure that you're recording from the correct muscle is to have the patient flex or supinate their forearm. So once you have set up your EMG measurement, what does your data look like? Here we see some raw data, or some unadulterated signal. What we are seeing here is moments of no electrical activity when the line is flat, then bursts of electrical activity when the muscle is activated. The signal has both positive and negative values that reflect the changes in voltage that are being measured. So how do we go from this signal to a signal that's informing us about what the muscle is doing? This is done through what we refer to as post-processing of the data. The commonly used steps of post-processing are as follows. First, in the graph that we're seeing here, every time that we see a negative value in the original raw EMG signal, we turn it into a positive value. The signal that we create from doing this is called a full-wave rectified signal. By doing this, we create a new plot that shows us just positive values that will correspond nicely with those points in time when the muscle is activated. Next, we filter or smoothen out the data. I will explain this process in Lecture 6 when I talk to you about frequency filters and moving average smoothing techniques. This step is referred to as performing a linear envelope. This step removes the erraticness of the data and allows you to get a clearer picture of what's going on with the muscle activation. So once you've performed these post-processing steps on the data, what does that leave us with? Using the post-process signal, we can measure things like the moment at which activity starts in the muscle, and the time of and level of peak muscle activity. We can also measure things like the duration of muscle activity. Okay, this completes our third lecture looking at the biomechanics of muscles.